Good morning, everyone. Today we are going to explore a very exciting and rapidly evolving field in biotechnology, the use of biological systems to generate sustainable energy, particularly through algae. As biotechnology students, you have already studied the fundamentals of metabolism, bioengineering, and microbial systems. In this session, we will integrate that knowledge into the real-world application of bioenergy. Bioenergy, in simple terms, refers to energy derived from biological sources, and algal biotechnology plays a major role in advancing this area. Algae are fascinating organisms. They are highly efficient photosynthesizers that convert sunlight, carbon dioxide, and nutrients into biomass rich in lipids, carbohydrates, and proteins. These biomolecules can be converted into fuels, feed, and valuable chemicals. Over the next few slides, we will examine how bioenergy works, why algae are such a powerful resource, and what challenges and opportunities lie ahead. My goal is not just to introduce concepts, but also to help you think critically about how biotechnology can contribute to sustainable energy systems in the future. Let us start by understanding what bioenergy actually means. Bioenergy is energy that comes from materials of biological origin, things like plants, agricultural residues, forest waste, and even microorganisms. It is an essential part of the renewable energy portfolio because it provides a carbon-neutral or even carbon-negative alternative to fossil fuels. This is particularly important in the context of global climate change, where we are trying to reduce greenhouse gas emissions while still meeting growing energy demands. Bioenergy is produced in several forms, for example, solid biomass that can be burned directly, biogas generated through anaerobic digestion, bioethanol from fermentation of sugars, and biodiesel produced from oils or fats. Each of these has different technological requirements and efficiency levels. The main reason bioenergy is so important is that it allows us to close the carbon loop. The carbon dioxide released during fuel combustion is roughly equal to the amount absorbed by the organism during its growth. In addition to energy, bioenergy systems support waste management and rural development. However, traditional bioenergy crops compete with food production and require arable land and freshwater. This is where microbial sources, especially algae, offer significant advantages, which we will discuss in more detail shortly. When we look at the sources of bioenergy, we can categorize them into three major groups: plant-based, waste-based, and microbial-based systems. Plant-based sources include crops like corn, sugarcane, and switchgrass, which are rich in starch, sugar, or cellulose. These are excellent for producing bioethanol or solid biomass fuels. However, the cultivation of such crops demands vast land and water resources and can sometimes interfere with food security. Waste-based sources, such as agricultural residues, forest byproducts, and even municipal solid waste, are gaining attention because they utilize materials that would otherwise be discarded. These wastes can be digested anaerobically to produce biogas or processed thermochemically to yield syngas and biooil. The third and perhaps most promising group is microbial-based systems, which include bacteria, fungi, and particularly algae. Algae stand out because of their high productivity, fast growth, and ability to grow in non-arable areas, such as saline lakes or wastewater ponds. When comparing all these sources, microbial systems often exhibit higher yield per hectare and lower environmental footprint. However, the technologies to cultivate, harvest, and process these organisms are still developing. As we move through this lecture, we will focus primarily on the algal systems, which are emerging as the next generation of sustainable bioenergy producers. When we compare different sources of bioenergy, it is important to look beyond the type of organism or feedstock and evaluate them based on measurable performance indicators, mainly their energy yield, production cost, and overall carbon footprint. This table summarizes those factors to give us a more holistic view of their sustainability and practicality. If we start with first-generation bioenergy crops like corn and sugarcane, they have relatively high energy yields, between 15 and 20 megajoules per kilogram of biomass, and are supported by well-established processing technologies. However, they often compete with food production and rely on intensive farming, leading to moderate carbon emissions and higher land use. Sugarcane, particularly in tropical regions, tends to perform better due to its efficient photosynthesis and the ability to use its byproduct bagasse for additional energy generation. Moving to second-generation sources such as switchgrass and agricultural residues, these materials offer slightly lower yields but are much more sustainable because they require fewer inputs and utilize non-food biomass. Their carbon footprint is lower, especially when the residues are used instead of being burned or discarded. Biogas, produced through anaerobic digestion of organic waste, is particularly attractive because it recovers energy from materials that would otherwise release methane, a potent greenhouse gas. Now, when we look at microalgae, the numbers become impressive in terms of energy potential, reaching up to 35 megajoules per kilogram of dry biomass. Algae grow extremely fast, capture large amounts of carbon dioxide, and do not require arable land. The major drawback is cost, mainly due to cultivation, harvesting, and drying processes, but technological improvements are gradually reducing these barriers. Macroalgae, or seaweeds, also show promise for coastal systems, though their lipid content is typically lower. Finally, forestry waste remains a reliable renewable source, but it must be managed sustainably to avoid deforestation impacts. Overall, this comparison shows that no single source is perfect, each has its advantages and limitations. The future likely lies in integrated bioenergy systems, where different feedstocks are combined based on local resources, cost, and environmental goals. Algal biotechnology, in particular, stands out for its potential to deliver high-energy yields with a low carbon footprint, especially when coupled with waste utilization and carbon capture strategies. Now that we have established the broader context of bioenergy, let us focus on algae, the microscopic powerhouses of sustainable biotechnology. Algal biotechnology involves using algae, both microalgae and macroalgae, for producing energy, food, feed, and other high-value bioproducts. Algae are a diverse group of photosynthetic organisms that thrive in a wide range of environments, freshwater, marine, and even extreme habitats. Microalgae, such as chlorella, spirulina, and nanochloropsis, are unicellular and grow rapidly, while macroalgae like ulva and laminaria are multicellular seaweeds visible to the naked eye. What makes algae particularly attractive for bioenergy production is their extremely high photosynthetic efficiency, sometimes several times higher than terrestrial crops. They can convert sunlight, carbon dioxide, and nutrients into large amounts of biomass containing lipids, carbohydrates, and proteins. Another major advantage is that algae do not compete with food crops for arable land or freshwater. Many species can grow in saline or wastewater environments. Additionally, they can help capture carbon dioxide emissions from industrial sources, integrating environmental remediation with fuel production. Algal biotechnology, therefore, sits at the intersection of renewable energy, environmental engineering, and molecular biology, making it an exciting field for biotechnologists to innovate in. To produce bioenergy efficiently, algae must be cultivated in systems that allow for optimal growth, resource use, and biomass productivity. Broadly, we use two types of cultivation systems: open ponds and closed photobioreactors. Open ponds are the simplest and most cost-effective systems, typically consisting of shallow raceway ponds that are continuously mixed to keep the algae suspended and exposed to sunlight. These are suitable for large-scale, low-cost production, but face challenges like contamination, evaporation losses, and less control over growth conditions. On the other hand, photobioreactors, often abbreviated as PBRs, but I will refer to them fully, are enclosed systems made of glass or transparent plastic tubes that allow precise control of light, temperature, carbon dioxide supply, and nutrient levels. While they are more expensive to build and maintain, photobioreactors produce higher biomass yields and are ideal for high-value or genetically modified strains. The main factors influencing algal growth include light intensity and wavelength, temperature, pH, carbon dioxide concentration, and nutrient availability. especially nitrogen and phosphorus. Understanding how these parameters interact allows us to engineer systems that maximize productivity while minimizing cost. Recent developments also include hybrid systems that combine the advantages of both open and closed systems. As future biotechnologists, your role will be to innovate cultivation strategies that are both economically viable and environmentally sustainable. Once algae are grown successfully, the next challenge lies in converting that biomass into usable energy or bioproducts. The process of algal biomass production typically involves several key stages: cultivation, harvesting, drying, extraction, and conversion. Cultivation, as we discussed, is where the algae grow and accumulate valuable compounds. After sufficient growth, we move to harvesting, separating the algal cells from the growth medium. This step can be done through flocculation, centrifugation, or filtration. Because algal cells are small and suspended in large volumes of water, harvesting often represents one of the most energy-intensive parts of the process. After harvesting, the biomass is usually dried to remove moisture. Common drying methods include solar drying, spray drying, or freeze drying, depending on the scale and product requirements. Once dried, we extract the valuable biomolecules, mainly lipids for biodiesel, carbohydrates for bioethanol, and proteins for feed or biofertilizers. The extracted lipids can then be converted into biodiesel through transesterification, while the remaining biomass can be fermented or anaerobically digested to produce bioethanol or biogas. The efficiency of this entire chain, from cultivation to conversion, determines the economic feasibility of algal bioenergy. Modern research aims to integrate these steps into continuous systems that minimize waste and maximize overall energy output, forming the basis of a sustainable algal biorefinery. Now, let us discuss how algae are transformed into different types of biofuels. Algae are remarkable because they can produce all the major classes of biofuels: biodiesel, bioethanol, biogas, and even biohydrogen. The most prominent of these is biodiesel, derived from algal lipids or oils. The process involves extracting the lipid fraction from algal biomass and then performing a chemical reaction called transesterification, in which the oils are converted into fatty acid methyl esters, the actual biodiesel, and glycerol as a byproduct. Algal species such as Nanochloropsis, chlorella, and Cenodesmus are known for their high lipid content, sometimes reaching over 50% of their dry weight. Besides biodiesel, algae can also produce bioethanol from their carbohydrate fraction. The starch or cellulose components can be hydrolyzed into sugars and fermented by yeast to yield ethanol. The residual biomass that remains after lipid extraction still holds value. It can be anaerobically digested to produce biogas, primarily composed of methane and carbon dioxide. Some cyanobacteria and green algae can also generate biohydrogen through photobiological processes, using sunlight to split water molecules. When we compare these fuels in terms of energy content, biodiesel typically provides the highest yield per unit mass, but each form of fuel has its own advantages depending on the intended application. The real promise lies in integrating all these processes within one algal biorefinery, where every component of the biomass is utilized efficiently, minimizing waste and maximizing energy output. In this table, we are comparing the main types of biofuels that can be produced from algae, their energy yields, and their overall conversion efficiencies. Let us go through each type to understand both their potential and limitations. Starting with biodiesel, this is produced by extracting lipids from algal cells and converting them through transesterification into fatty acid methyl esters. Biodiesel has a high energy yield, roughly 35 to 40 megajoules per kilogram, which makes it comparable to petroleum diesel in terms of energy content. Its conversion efficiency ranges from 40 to 55%, depending on the species and extraction method. Biodiesel is currently the most commercially advanced algal biofuel because it integrates well with existing diesel engines and infrastructure. Next, bioethanol is derived from the carbohydrate fraction of algae. Carbohydrates such as starch or cellulose are fermented by microbes to produce ethanol. Its energy yield is slightly lower, around 25 to 30 megajoules per kilogram, and the conversion efficiency is typically between 30 and 45%. While ethanol has a lower energy density than biodiesel, it is compatible with gasoline engines and can be blended with fossil fuels. Technological challenges in efficiently converting algal carbohydrates are being addressed through enzymatic and microbial engineering. Biogas is produced by anaerobic digestion of the residual algal biomass remaining after lipid or carbohydrate extraction. This process generates a methane-rich gas with an energy yield of approximately 20 to 25 megajoules per kilogram. Conversion efficiency is higher, ranging from 50 to 65%, because it uses materials that would otherwise be considered waste. Biogas is particularly important in integrated biorefineries, where every fraction of biomass contributes to energy production. Finally, biohydrogen is generated through photobiological or photofermentative processes, where certain algae or cyanobacteria use sunlight to split water molecules or ferment organic matter into hydrogen. Although hydrogen has an extremely high energy content per kilogram, over 100 megajoules, the overall conversion efficiency is currently very low, around 5 to 15%. Despite its low efficiency, biohydrogen represents a highly promising renewable fuel for the future if technical hurdles can be overcome. Overall, this table illustrates that no single biofuel pathway captures all the energy in algal biomass. A biorefinery approach, where lipids, carbohydrates, and residual biomass are all utilized, maximizes energy recovery and minimizes waste, making algal biotechnology a highly versatile platform for sustainable energy production. A crucial frontier in algal bioenergy research is the use of genetic engineering to enhance productivity and resilience. Traditional strain selection can only go so far, so scientists are now using molecular tools to directly modify algal genomes. Techniques such as CRISPR, Cas9, homologous recombination, and transformation with engineered vectors have opened new possibilities for strain improvement. The main objectives of genetic modification in algae include increasing lipid accumulation, improving carbon dioxide fixation rates, and enhancing tolerance to stress factors such as high salinity, temperature fluctuations, or nutrient deprivation. For instance, metabolic engineering can redirect carbon flux from carbohydrate synthesis toward lipid biosynthesis, resulting in much higher oil yields. Similarly, overexpression of certain enzymes in the photosynthetic pathway can boost light utilization efficiency. There are even efforts to engineer algae to secrete oil directly into the medium, which would make harvesting far simpler. A well-known example is the modification of Nanochloropsis species to produce higher levels of triacylglycerols, the main precursors of biodiesel. Beyond energy, genetic tools also enable the production of high-value compounds such as pigments, antioxidants, and omega-3 fatty acids. However, it is important to consider biosafety regulations and public perception when working with genetically modified algae, especially for open environment cultivation. As biotechnologists, it is essential to balance innovation with ethical and environmental responsibility. One of the strongest arguments for investing in algal biotechnology is its potential to deliver simultaneous environmental and economic benefits. Unlike traditional energy crops, algae can grow on non-arable land and use saline or wastewater, meaning they do not compete with food production or freshwater resources. They also act as effective carbon sinks. During photosynthesis, algae capture carbon dioxide from the atmosphere or directly from industrial flue gases, converting it into biomass. This not only reduces greenhouse gas emissions, but also contributes to carbon neutrality. Additionally, algae can remove nutrients such as nitrogen and phosphorus from wastewater, helping to prevent eutrophication of natural water bodies. On the economic side, the use of residual biomass for producing fertilizers, animal feed, and other co-products makes the entire system more profitable. When combined with wastewater treatment and carbon capture, the economics of algal bioenergy improve significantly because you are solving multiple environmental problems while generating valuable products. The scalability of such systems still depends on local infrastructure, energy policies, and investment support, but pilot projects around the world have shown that when integrated properly, algal systems can form the foundation of a circular bioeconomy, one where waste becomes a resource and sustainability is built into the production cycle. These advantages make algae a unique and highly versatile platform for the next generation of green technologies. When evaluating bioenergy options, it is essential to compare algae with traditional terrestrial crops, as they differ significantly in productivity, sustainability, and environmental impact. Starting with growth rate, algae can double their biomass in just 1 to 2 days under optimal conditions, while terrestrial crops such as corn, sugarcane, or switchgrass typically require months to reach harvest maturity. This high growth rate is one of the reasons algae achieve much higher energy yields per hectare, sometimes 20 to 50 times more than conventional crops. Another important factor is land requirement. Algae can grow on non-arable land, in brackish water, or even in wastewater systems, meaning they do not compete with food crops. In contrast, terrestrial bioenergy crops require fertile, arable land, which often leads to conflicts with agriculture and food production. Similarly, water requirements are much lower for algae if saline or wastewater sources are used, whereas terrestrial crops often need extensive irrigation, increasing pressure on freshwater resources. Photosynthetic efficiency is also significantly higher in algae, reaching up to 5 to 10%, compared to 1 to 3% in most land crops. This translates into a much higher biomass and energy yield per area. Regarding environmental benefits, algae can sequester carbon dioxide directly from the air or from industrial flue gases, and can also help remediate wastewater. While terrestrial crops provide moderate carbon capture and may lead to soil degradation, nutrient runoff, or pesticide issues. Finally, while harvesting and processing algae is currently more complex and energy-intensive, terrestrial crops benefit from well-established agricultural infrastructure. Overall, algae represent a highly sustainable and productive alternative for bioenergy, complementing terrestrial crops and offering a pathway toward a low-carbon, circular bioeconomy. While algae hold immense promise as a renewable energy source, it is important to recognize the significant challenges that still limit large-scale commercialization. The first and most critical issue is economic viability. Cultivating algae, especially in closed photobioreactors, remains costly due to high energy and infrastructure demands. Harvesting and drying also consume large amounts of energy because of the dilute nature of algal cultures, typically less than 1 gram of biomass per liter of water. These steps can account for up to 30 to 50% of the total production cost. Another challenge is biological variability. Algal growth and lipid productivity are strongly affected by environmental conditions such as light, temperature, and nutrient levels, which makes maintaining consistent yields difficult. Contamination by unwanted microorganisms is another major problem, particularly in open pond systems. In addition to technical barriers, there are policy and market-related obstacles. Subsidies for fossil fuels, lack of carbon pricing mechanisms, and insufficient public awareness slow down the transition to algal-based bioenergy. Despite these hurdles, ongoing research focuses on improving algal strains through genetic engineering, optimizing cultivation systems, and integrating processes to recover multiple products, an approach known as the biorefinery model. Governments and private companies are increasingly investing in pilot projects to address scale-up and cost reduction challenges. As researchers, our task is to bridge the gap between laboratory success and industrial reality, ensuring that algal bioenergy becomes both sustainable and economically competitive. The biorefinery concept is central to the future of algal biotechnology. Much like a petroleum refinery that produces multiple fuels and chemicals from crude oil, a biorefinery processes biomass to generate a spectrum of valuable products, fuels, chemicals, food ingredients, and fertilizers. In the case of algae, this integrated approach is essential because producing a single product, such as biodiesel, rarely covers the high costs of cultivation and processing. By utilizing every component of the algal biomass, we can achieve economic and environmental sustainability simultaneously. For instance, lipids can be converted into biodiesel, carbohydrates can be fermented to bioethanol, proteins can be used for animal feed or biofertilizers, and pigments like chlorophyll or beta-carotene can be sold as nutraceuticals or cosmetic ingredients. Additionally, residual biomass after extraction can undergo anaerobic digestion to produce biogas, thus recycling nutrients and generating energy for the process itself. This zero-waste philosophy embodies the principles of the circular bioeconomy. The integration of carbon capture, wastewater treatment, and energy recovery makes algal biorefineries a highly sustainable model. Some advanced facilities also couple algae cultivation with flue gas from industries, simultaneously reducing emissions and generating biomass. In the future, modular biorefineries could be established near industrial zones or wastewater treatment plants, making bioenergy production decentralized and efficient. The biorefinery model thus represents the most promising pathway for scaling algal biotechnology to industrial levels. To better appreciate how these concepts are applied in the real world, let us look at a few significant case studies from around the globe. In India, the Council of Scientific and Industrial Research, specifically the National Institute for Interdisciplinary Science and Technology, developed a pilot-scale microalgae-based biodiesel project. This system utilizes indigenous algal strains and local wastewater, demonstrating the feasibility of decentralized fuel production in tropical regions. In the United States, the National Renewable Energy Laboratory has been leading algal biofuel research for decades, developing efficient photobioreactor designs and exploring genetic improvements in lipid production. Their studies have significantly improved the understanding of large-scale algal cultivation. Meanwhile, in Japan, the company Euglena Company Limited has successfully commercialized biojet fuel from the microalga Euglena gracilis, partnering with airlines to conduct demonstration flights. These case studies highlight how algae-based bioenergy systems can adapt to different national contexts and technological strategies. Each project addresses local challenges, whether it is resource availability, climate, or infrastructure, while contributing valuable insights into process optimization and scalability. Such global initiatives also underline the importance of interdisciplinary collaboration among biologists, engineers, policymakers, and industry leaders. The lessons learned from these efforts are paving the way toward making algal bioenergy a realistic and sustainable part of our global energy portfolio. As we look toward the future, the potential of algal biotechnology appears both vast and transformative. The field is rapidly evolving due to advances in synthetic biology, systems biology, and process engineering. One of the most promising directions is the use of synthetic biology to redesign algal metabolic pathways for higher efficiency. By precisely editing genes, we can create strains that fix carbon dioxide more effectively, accumulate higher lipid content, or even produce entirely new bio-based molecules. Another exciting area is the integration of algal systems with industrial carbon capture. Imagine coupling algae cultivation units with power plants or cement factories, using their flue gases as a source of carbon dioxide. This not only mitigates emissions, but also provides free feedstock for algal growth. Wastewater-based algal cultivation is also gaining traction, offering a dual benefit: nutrient removal and biomass production. Moreover, hybrid systems that combine photobioreactors with open ponds can optimize both cost and yield. On the technological side, automation, artificial intelligence, and data analytics are being employed to monitor and control algal growth in real time, improving process stability and reducing costs. Policy and economic frameworks are equally important. International collaborations and green finance mechanisms are now recognizing algae as part of the broader circular bioeconomy. In the coming decades, we can expect algae to contribute not only to biofuels, but also to bioplastics, pharmaceuticals, and carbon-negative materials, making it a cornerstone of sustainable biotechnology. Let us conclude by revisiting the key ideas we have explored in today's session. Bioenergy represents a renewable, sustainable approach to meeting the world's growing energy demands, and among the various sources available, algae stand out as one of the most promising. Their high photosynthetic efficiency, rapid growth rate, and ability to thrive on non-arable land make them ideal for large-scale energy production without compromising food security. We examined how algae can be cultivated in both open and closed systems, harvested and processed to produce multiple types of biofuels, including biodiesel, bioethanol, biogas, and biohydrogen. Through genetic engineering, we can further enhance their productivity and resilience, moving closer to industrial-scale feasibility. We also discussed the environmental and economic benefits, such as carbon capture, nutrient recycling, and integration with wastewater treatment. However, significant challenges remain, particularly in cost reduction, scalability, and regulatory frameworks. The biorefinery model emerged as a key solution, allowing us to extract multiple products from a single biomass source and achieve true circularity. Finally, global case studies from India, the United States, and Japan have demonstrated that the transition from research to application is already underway. As future biotechnologists, your role will be to push this frontier forward, innovating processes that make algal energy not only possible, but practical and sustainable for generations to come. Thank you for listening. I will see you in the next lecture.